The rate of photosynthesis is a crucial aspect of plant biology, directly influencing the growth and productivity of plants. It can be affected by several environmental factors, each of which can be represented in a graph that illustrates its impact on the process. Understanding these graphs and the underlying principles helps in optimizing conditions for plant growth, especially in controlled environments like greenhouses. The first important graph related to photosynthesis shows the relationship between light intensity and the rate of photosynthesis. Photosynthesis requires light energy, and as light intensity increases, the rate of photosynthesis also rises. However, this increase is not indefinite. At low light intensities, the rate of photosynthesis is limited because there is insufficient energy to drive the process effectively. As light intensity continues to increase, the rate of photosynthesis rises sharply until it reaches a plateau. At this point, other factors, such as carbon dioxide concentration or temperature, become the limiting factors, and further increases in light intensity do not enhance the rate of photosynthesis. This plateau indicates that the chloroplasts within the plant cells are fully saturated with light, and any additional light does not result in a faster rate of photosynthesis. The second graph illustrates the effect of carbon dioxide concentration on the rate of photosynthesis. Carbon dioxide is a key reactant in the photosynthetic process, and its availability directly influences the rate at which glucose is produced. When carbon dioxide levels are low, the rate of photosynthesis is also low because there is not enough of this raw material to sustain the process at a high rate. As the concentration of carbon dioxide increases, the rate of photosynthesis increases as well, until it reaches a saturation point where the rate levels off. Beyond this point, even if more carbon dioxide is available, the rate of photosynthesis will not increase further because other factors, such as light or temperature, may now be limiting. The third graph represents the effect of temperature on the rate of photosynthesis. Photosynthesis is an enzyme-driven process, and temperature plays a critical role in enzyme activity. As the temperature rises, the rate of photosynthesis generally increases because the enzymes involved in the process work more efficiently. However, this is only true up to an optimum temperature, usually around 25 to 30 degrees Celsius for many plants. Beyond this optimal range, the rate of photosynthesis begins to decline because the enzymes start to denature, losing their functional shape and ability to catalyze reactions. If the temperature drops too low, enzyme activity slows down, reducing the rate of photosynthesis. These three factors, light intensity, carbon dioxide concentration, and temperature, do not operate in isolation. Often, one graph may show the effect of multiple limiting factors at once, illustrating how they interact to influence the rate of photosynthesis. For example, even if light intensity is high, a lack of carbon dioxide or suboptimal temperatures can still limit the rate of photosynthesis. Similarly, an increase in carbon dioxide will only boost photosynthesis if there is enough light and the temperature is within the ideal range. One way to measure the rate of photosynthesis is by observing the production of oxygen, which is a byproduct of the process. By measuring the amount of oxygen produced over a certain period, scientists can determine how quickly photosynthesis is occurring. This method is often used in laboratory settings to assess how different factors influence the rate of photosynthesis. The relationship between light intensity and distance from the light source is governed by the inverse square law, which states that the intensity of light is inversely proportional to the square of the distance from the source. This means that as the distance from the light source increases, the light intensity decreases rapidly. For example, if the distance is doubled, the light intensity falls to a quarter of its original value. This principle is crucial in understanding how light affects photosynthesis, particularly in agricultural settings where artificial lighting is used. To maximize the rate of photosynthesis, especially in farming, it is possible to artificially create ideal conditions. In greenhouses, for instance, light intensity can be controlled with artificial lights, carbon dioxide levels can be enhanced, and temperature can be regulated to maintain the optimal range for photosynthesis. By managing these factors, farmers can ensure that plants are photosynthesizing at their maximum potential, leading to higher yields and more efficient crop production. This controlled environment allows for the fine-tuning of conditions that would be impossible to maintain in an open field, making it a valuable technique for modern agriculture.